I think we need to move on to the next talk, which is from Tim Storr at Simon Fraser University in Canada. He is professor and chair of the graduate program in chemistry there. And his interest is in medicinal inorganic chemistry and catalysis, bioorganic chemistry, and in neurodegenerative disease, in particular, developing therapeutic agents. He's interested in bimetallic cooperativity. Today, his talk is entitled Using Multifunctional Molecules for Therapeutic Intervention of Mutant P53. Tim.
Thank you very much for the introduction. And again, thanks to RAMS for putting together this really neat seminar. I'm taking us off in a little bit of a different direction, again, looking at the P53 protein in cancer, but you'll see the commonalities in terms of zinc binding and also amyloid aggregates that form with this protein. And so this is work done here at SFU, my graduate students and myself, as well as with Christian Guédon, who's in Strasbourg. And so he's been a big component of this work as well. And so as we all know, protein misfolding and aggregation play a significant role in disease, and this can be due to small changes in primary structure. Even a point mutation, as you'll see in this talk, can have structural and functional consequences. And of course, this is of relevance to neurodegeneration in terms of Alzheimer's disease, Parkinson's disease, ALS, and TDP-43. We heard many talks in the middle section about diabetes and IAPP, but I'm going to focus in on cancer and the P53 protein. And so the P53 protein has been affectionately called the guardian of the genome. And in response to cancer phenotypes such as oncogene activation, DNA damage, and hypoxia, activation pathways are ongoing in cells, and P53 is expressed. It forms a tetramer. And then basically you have target gene transcription. And following that, you either get apoptosis, DNA repair, cell cycle arrest, or cellular aging. And so this is very important for the body to combat cancer. However, cancer is smart, cancer is evolved, and P53 is inactivated in almost all tumors. And so there's a big focus on if we can develop small molecules or intervene in this pathway to reactivate the P53 protein so that it can do its job. And so with this P53 protein, over 50% of cancer diagnoses involve mutations directly to P53. And so here's the P53 protein, and particularly the DNA binding domain. So in this DNA binding domain, we have a number of point mutations which are relevant to a lot of different cancers. And mutations lead to a loss of function due to limited DNA recognition. I'll show you a structure in a moment, and also lower stability. And this leads to unfolding and aggregation, amyloid formation, and also loss of a structural zinc. And so here is a crystal structure of the protein, at least the DNA binding portion, bound to a DNA here. You can see a variety of these point mutations which are significant in different cancers. Some of these are DNA contact mutations. Some of them center around that structural zinc. This is a zinc finger motif where that one structural zinc is bound and is obviously of critical importance to folding and the overall structure of the protein. There's other ones that are related to unfolding and stability, and we've actually focused, as you'll see today in this talk, on this Y220C mutation. It's remote from DNA binding and also the zinc binding portions. However, one can stabilize, there's a pocket that forms here, and if one can stabilize this region, you can also stabilize the overall protein and obviously have it do its job. And just an example of aggregation, protein only in the absence of obviously its structural other components. You see aggregation over time, just based on THT fluorescence. With EDTA addition, you see almost instantaneous aggregation of the protein. So a loss of zinc leading to unfolding and aggregation and beta sheet formation. And so aggregation leads to a loss of protein function. And obviously this is very important for obviously if we can interact and interject here and keep this protein in its native folded state. And so here's a cartoon of zinc binding to P53. It's obviously essential for DNA binding and proper protein folding. And so under normal conditions of zinc, we have folding, we have tetramization, and we have specific DNA bonding. And so this is how P53 is working in its equilibrated state. If we have insufficient zinc, we can then get to apo P53, which then has tetramization potential, but leads to non-specific DNA binding and obviously promoting aggregation. And then with excess zinc, we have obviously the same endpoint, which is aggregated P53 and the inability to fold and sense DNA in the cell. And so there's a number of areas for therapeutic intervention, especially for small molecules. Here is obviously the native folded P53 with its structural zinc binding to DNA. And then we have transcription to go on to undergo cell cycle arrest, DNA repair of apoptosis. And so people and researchers have developed molecules to try and keep high levels of P53 in cells by interacting with MDM2, which basically functions to degrade P53. And also, there's a number of research groups out there who have now tried to look at molecules that will shuttle zinc and re-equilibrate zinc into the cell to try and refold P53, stabilize conformations by binding to P53, and then also over here, and of relevance to today's talk, preventing aggregation by keeping it in its monomeric state. And so there's a number of references at the bottom here for other groups who have looked at P53 amyloid aggregation and what it means in terms of function and combating cancer. And so P53 stability has been a primary target. Is it possible to bind small molecules to P53 to allow it to function in its normal state? Wilkin and Furst and others have developed molecules that specifically target a pocket on the Y220C mutant that I identified earlier. These phenols here, which stabilize the protein in a remote site and allow it to continue its normal function. Some really interesting work by Dennis Carpizzo and Stuart Lowe in the United States, looking at these simple thiosemcarbozones, which have specific KD values for zinc and are able to shuttle zinc into the cells from media and obviously in the body as well. And then they can also show that they have increased folding and stabilization of the P53 molecule. And so we came at this. We'd done a lot of work in amyloid and in Alzheimer's disease, and we thought about this from the perspective of, could we actually transfer some of the molecules that we know work in terms of interacting with amyloids and also interacting with zinc and copper in these models to the P53 protein. And so conceptually, we developed a series of molecules with these iodinated phenols for binding into the Y220C pocket and also a zinc chelation moieties. And so could we effectively develop molecules that will shuttle zinc into the cells and also stabilize this Y220C mutant? And so here is a series of 7 molecules that we published on just to show some of our first generation derivatives. We've moved on from these now in work we're doing at the moment, but I'll structure this talk in terms of these first 5 was really the first publication, and then the second 2 was a later publication in this area. So we measured the ligand zinc KD values in nanomolar here, so you can see this across. So these are essentially in the low nanomolar range for zinc binding. Do note here that the zinc KD of P53 is approximately 2 nanomolar. However, these point mutations will change it slightly. And so for example, Y220C is a slightly lower, a weaker KD for zinc binding. And so in initial screens at the NCI 60, which is a panel of cancer cell lines, we saw that L1 to L3, these ones here, did not show in vitro cytotoxic activity in the NCI 60 screen. However, L4 and L5, these two here, and LI and LH were cytotoxic and also have appropriate zinc KD values to act as metallochaperones. And so these are the ones that we really took forward into further studies to really understand their mechanisms of action. And so here is a metallochaperone study looking at increasing intracellular levels of zinc in the P53. So this is the Y220C cell line. And so essentially we incubated with the FZ3AM. So this is the zinc specific fluorophore. In the presence of added zinc chloride to media, we see really no change in zinc intracellularly. If we add in the perithione here, which is a positive control for zinc uptake, you can see significant amounts of zinc now in the cell. And then with L4 and L5, you also see an increase. And especially for L5, L5, we see approximately a 4-fold increase in zinc intracellularly in compared to no treatment control. So this is quite promising for this L5 molecule and its ability to bring zinc into these cancer cells. We then looked at the toxicity more specifically. And so in a 3D spheroid model, the Y220C cell line, we see essentially the same or increased activity of L5 in comparison to oxaliplatin. And so you can see the IC75s there. I just see your videos here. I'll bring this down a bit of 19.5 for the L5, which is again, increased in comparison to oxaliplatin. So we're seeing significant cytotoxicity. That's useful. What we also saw was target gene upregulation. And so this is again showing that we have some specificity for the P53 pathway. And so at the 24-hour time point after addition of our compound, you can see here no upregulation in actual P53 concentrations, but we are seeing upregulation in apoptosis gene markers and also in the P21 cell arrest marker, which is a canonical marker for P53 activation. And so this is showing that we can get zinc into cells and that we can also see this increase in activation of P53 target genes. So this was quite a promising result for us in this with this initial series of molecules. And just to say a little bit more, we looked at some docking studies, and indeed these compounds do dock into that same pocket of the Y220C mutant, mirror those for known stabilizers. And then we also looked at immunoprecipitation in this cell line, and we see decreased unfolded P53 with L5 treatment. And that's just showing unfolded P53 here. However, in the presence of 15 and 30 micromolar of the L5 compound, we're seeing less unfolded P53 in these experiments. So again, showing that we are likely increasing the folded component of P53 and increasing the activation of P53 without increasing overall concentration. And so, again, specific to this symposium is really looking at aggregation of peptides and proteins and the role of metal ions in this. And so it is quite commonly known that the lower stability of P53 mutants leads to protein unfolding and aggregation. And so these mutants can co-aggregate with wild type P53, and thus this is an important therapeutic target. And so looking at the 282 mutant, this is in cells, you can see here in red, this is all the mutated and aggregated P53 in these cells in the cytoplasm. So again, significant aggregation of P53. Again, due to its overexpression, this mutant isn't doing what it's supposed to do. And so the cells are increasingly basically bringing out more and more of this P53 and try to affect its job. And so Eisenberg and coworkers developed this peptide here, which was designed specifically to bind to amyloid aggregates of the P53 protein. And they showed in these two cell lines, the R175H and R273 mutants, that it could restore P53 function. And also you can see here in terms of tumor volume, vehicle and the scrambled peptide, we see no change. They say it saw no change in P53 function. They also saw no change in tumor volume. However, with their peptide, you can see this decrease. So again, showing these aggregation inhibitors could actually affect tumor volume and really restore P53 function in these mutants. And so again, we were interested in inhibiting mutant P53 activation, and particularly the Y220C cell line, as we'd already initiated our development of molecules for this specific mutant. We knew from previous work by Silva and also First that P53 aggregates with Y220C did form, and you can see this by THT fluorescence here. So again, showing this common assay for fibrillization of proteins. And so we developed a series of molecules shown here with THT-like analogs bound to a zinc chelate. And many of these mirror some of the molecules that Levieux just showed in a previous talk in terms of we knew what their zinc binding potential would be. And so we wanted to look at if these molecules would interfere with P53 aggregation and start to play a role in obviously basically getting this P53 back online. And so here are the two molecules. We looked at the binding using spectrophotometric titrations. Here are the complexes with zinc, and just showing you the KD values that we can calculate from our experiments for zinc binding to these two new ligands. And so again, showing their nanomolar affinity and of the approximate range to act as metallochaperones for P53 mutants in cells. And so we then looked at zinc uptake in much the same manner as I've already shown with the previous molecules. And we see approximately 3 and 2-fold increase in zinc in these cell lines in comparison to no treatment control. So again, we see an increase in zinc. So the metallochaperone activity is identified with these 2 molecules. We then looked at the aggregation profile of these molecules, again, by turbidity to start with, and we can see that there's a concentration-dependent effect with this Li molecule. And the only real difference between these 2 molecules is the iodine functionality ortho and hydrogen for the LH molecule. And we see at 5 and 10 micromolar Li, we see a decrease in the aggregation via turbidity with the P53 protein. However, for the LH molecule, we see essentially no change or no concentration dependent change. And so this is showing that the iodine version here is significantly slowing the aggregation rate. And here's a bar plot at the end of this study. We also looked at transmission electron microscopy. So no treatment at 30 minutes and 2 hours, we're essentially forming fibrils. However, if we add in our Li compound, we're seeing aggregates, but these are amorphous aggregates. And we might expect to see aggregates as this is an intrinsically disordered protein. And out of a cellular context, we'd start to see aggregation in this time frame. However, with LH, we're seeing fibrils. And so again, showing you this difference between even these 2 molecules with this iodine substitution. We then looked at cells and aggregation in cells rather than obviously just in a test tube. And here is the antibody for P53 under no treatment with 25 micromolar of our Li compound and then 25 micromolar of the LH compound. And you can see here with A11, which is an oligomer-specific antibody, looking at protein aggregation, we're seeing more aggregation under no treatment, and especially with L7, we're also seeing morphology changes, and this is indicating cytotoxicities you'll see at the end of this talk. But we're seeing a reduction in overall aggregates with the Li compound. Again, further confirmation that we are changing and at least reducing the rate of aggregation of P53 in this mutant cell line in a cellular context. And so this was quite exciting for us as well. And so looking at the binding interaction, we envisage is likely in the micromolar range. We had some difficulty actually measuring the binding interaction between these molecules and the protein based on ITC and fluorescence studies, just due to the limited solubility of these ligands that we had designed. We have our ideas for moving forward on this and increasing that binding specificity. But we were able to use native mass spectrometry with a colleague in Canada, Derek Wilson at York, to look at non-covalent interactions. And what we saw here's the mutant P53 envelope, the 9+ charge state. In the presence of our Li compound at 10 and 25 equivalents, we start to see adducts, non-covalent adducts for Li. So one molecule of Li and then 2 molecules of this Li ligand at that higher concentration. And what we did not see is these same adducts forming with the LH compound, again, showing us that this iodine is assisting in that interaction with the protein. And so moving to in vitro cytotoxicity. So again, we use the NCI screen, which is a free repository for looking at the anti-cancer activity of molecules. What we could see here is that the GI50s for both of the Li and LH compounds are 0.4 and 1.6 micromolar. And so this is quite promising. And so it went through a multi-dose study. But you can see here that there's a relatively broad spectrum toxicity in these cancer lines. And I'll say again something about that at the end of this talk. Unfortunately, in the NCI 60 database, there are no Y220C mutants, so we couldn't directly test that based on the NCI screen. So we then moved to a gastric cancer cell line. And so this is the second leading cause of cancer-related cell deaths. There's a cancer-related deaths in general. There's a 77% rate of P53 mutation in these cancers. So it really goes to show you the effectiveness of these of gastric cancers in really removing this ability of ours as humans in terms of defense. And so we tested in this gastric cancer cell line, and the Li and LH compounds, you can see here the IC50s at 4.5 and 24.6. So either comparable or better than cisplatin and oxaliplatin in this cell line. So this is again quite a promising toxicity, but toxicity isn't everything, and obviously it's the ability of these compounds to target cancer cells, which is really most important in comparison to normal cells. And so we then looked at the mechanism of action, right? And this is really important here, not to just stop at cytotoxicity. And so we looked at the ability of these compounds to induce apoptosis using an indicator protein, so caspase-3, which is cleaved during apoptosis. And so in a wild type cell line AGS, you can see here at either the IC50s or IC75s for oxaliplatin or our two candidate molecules, Li and LH, we see essentially no caspase or very little caspase activation in these cell lines. We do see upregulation of P53 though with oxaliplatin, and this is an interesting result, which is known for the P53 pathway. If we then go to the Y220C cell line, which is specific to the molecules we are developing, what we see here is we start to see caspase-3 activation for oxaliplatin and significant caspase-3 activation for the Li compound at the IC75 concentration. So again, promising result looking at the selective activation of caspase-3 for this iodinated construct that we developed. And so moving on to upregulation of P53 target genes. And so this is, again, really important if we want to say something about the P53 pathway and our ability to interact and modulate that pathway with this Li and the LH compound. And so we specifically went after P21, which is again, a marker of cell growth arrest and is really the canonical marker of P53 activation. And what we saw here, again, upon treatment with our compounds, we saw no changes in P53 activation. So this is important. So we weren't just increasing levels of P53. We wanted to ask the question of if we could say something about refolding or activation of the P53 that was present. And then we also did a knockout experiment, whereas if we knock out P53, what happens to these markers? And so what we see is upon addition of our two compounds, Li and LH, we see an increase in P53 activation via this P21 RNA expression. And so that's good. If we then knock this out, we see this decrease for Li. So this is indicating that we are decreasing the effectiveness of the P53 pathway. However, for this LH compound, we note here that we're still getting pretty much the same cell growth arrest for that derivative. And what this really suggests is that we're having P53 independent toxicity for this LH compound. And as in many cancer pathways, the P53 pathway is complicated. There's P63 and P73 proteins that are involved. And so there's many other facets to the pathway that, again, I've learned about in terms of our studies here, and my collaborator, Christian Guedon, has far more expertise in. And so again, this is showing this independence from the P53 pathway for the LH compound, but this P53 dependent activity for the Li compound. And so we're seeing this difference now in this toxicity, which is quite a useful series of experiments and information for us from these studies. And so we moved on, and part of our interest was, obviously the selective toxicity, and could we get selective toxicity in a cancer cell model versus a normal cell model? And this is always a problem. Cancer cells are fast dividing, normal cells are not, and so how does one develop a model which allows you to compare? And we moved to a small intestine organoid model. So these are three-dimensional multicellular structures, and looked at a long-term survival assay of our compounds. And here is no treatment. So here is the organoids, and you can see these well-developed structures that form, again, healthy organoids. If we add oxaliplatin, we start to see disorganization in terms of these membranes and dying and or dead organoids in this assay. And so we've sampled 200 to 400 organoids in each of these assays to come up with these bar plots here. And so in no treatment conditions, we essentially have mostly normal organoids. With oxaliplatin, we have disorganized, dying, and dead organoids. And with our Li compound, which is interesting to us, is we have actually significantly less toxicity in this non-cancerous model. And so we have normal and disorganized, also, of course, these dying and dead organoids. However, we are seeing significantly less toxicity here in comparison to oxaliplatin. And perhaps the most interesting was the almost complete cell death with this LH compound at this 10 micromolar concentration at 72 hours. Again, showing this P53 independent pathway, this widespread toxicity of this LH compound. And again, really showing how chemical design and simple structural changes can have this widespread change in overall toxicity in cancer cells versus non-cancerous models. And so we're moving on with these now, and especially with this LI compound into mouse models with this specific mutant. And we're also broadening our horizons to other cancerous mutants. And each of these mutants, each of these point mutations, and each of these proteins will behave in a different way and aggregate in a different way. And so we're broadening our horizons now in terms of the toxicity and cell killing ability of these of this LI compound and derivatives of this. And so the current data, as I mentioned, highlights the significance and the difference of an iodine functionality. Multiple modes of action are still likely here. However, this LI compound does show minimal toxicity in organoids and in vivo. So it's suggesting a specific P53 dependence, which we're really pleased about. And what's interesting about this is as one can now start to envision cancer cell lines and cancers where traditional agents such as chemotherapeutics, the platinum agents and so forth are known to be essentially inactive. If we can actually regain the function of P53, we can then regain the function of these common chemotherapeutics. And we see an example here where we have a possible synergism. So you can see here at 5 micromolar of our LI compound, we have a certain amount of caspase 3, but in the presence of oxaliplatin, which has no effect, and 5 micromolar of our ligand, we're now seeing significant caspase 3. So again, this is suggesting that we can actually sensitize these mutant cell lines and mutant cancer mutants to actually traditional chemotherapeutic drugs. And bringing us back to this copper and zinc debate that's occurred today, one of the most interesting parts of this is we are getting general ROS throughout these cell systems, and our thought process on this and others is that we are having non-specific copper binding and ROS generation, which is leading to some of this off-target toxicity. And so we are actually quite interested in minimizing copper and maximizing zinc binding in these molecules moving forward. And so with that, I'd like to summarize by thanking the group here. This is the group summertime, wintertime in Vancouver. One of my collaborators in the department at SFU, Jeffrey Warren. And this is Jessica Miller, my basically the first PhD student to work on this project. And she met Christian Guédon at a conference in Hawaii, which started the collaboration. So if you ever get a chance to send your students to Hawaii to a conference, please do so, as you never know what will come out of that. And Christian's been a fantastic collaborator on this project in particular. And so with that, there's been other people along the way who have helped with this project and are ongoing efforts in this area and these funding agencies. And so I'd be happy to answer any questions on this work.
Thank you very much, Tim. We have some questions. Let's start with a question from Peter Fowler. Do you have any direct evidence that the diiodophenol moiety binds to P53 in the cell, or increased binding to P53 compared to the chelator moiety only?
Yeah, so we see, so we've done a variety of different experiments, and I would say yes, compared to the chelator moiety only, we see increased interactions. Again, we're in a low micromolar range with fairly low water solubility for these ligands. So it precludes a lot of kind of standard methods to look at interactions. And we're moving to 2D NMR studies to try and get a better KD value for these interactions and to actually map out specific protein sites for these interactions. And so, yes, we do see an increased binding compared to the chelator. I think, however, we can improve those interactions. But realistically, we want to broaden to other mutants. As I showed at the start, there's a number of point mutants, and we picked the one that's, I think, the 10th most likely. I really like to bring us back to the R175H or the 282 mutants, which really are the most prominent mutants for P53 in different cancers.
Okay, I think we have time for maybe one more question from Anup Arunagiri. Nice work, Tim. 2 questions for you. 1, with the introduction of cis Y220C cause P53 intracellular misfolding involving disulfide-linked intermolecular interactions? And I'll tell you the second one after you answer that one.
Okay, I think that's likely a possibility. It's that hasn't been studied. There's some recent work by Stuart Lowe, who's done some really nice, very careful studies looking at zinc binding in these mutants, and the structural consequences of that. And one of the interesting points that's come up is actually this cysteine mutation actually starts to compete for zinc. And it may be the additional binding of zinc in that cavity that's also causing some complications. So again, another reason for trying to block that with small molecules. But there's a number of Y220C and Y220X, if you will, amino acid mutants. And the increased aggregation isn't specifically for the cysteine mutant, but I think that would likely play at least some role in this cell. So it's a good question.
Okay, so the second question from him is, how is P53 folding affected in gastric cancer cells? If this is related to mutations, are these mostly located in the P53 DNA binding domain in this case? But I think you've answered that already, one.
Yeah, different cancers have different mutations,
Yeah.
And I think I've shown the point mutations. I don't have the exact point mutation changes for gastric cancer. That plot at the start is kind of effective over all cancers that have been measured. But I think there's a variety of mutations in gastric cancers, and this really comes down to our ability to get to personalized medicine and really genome mapping of specific phenotypes in patients to come up with the best therapies that we have available.
Okay. And one last question from me. I was wondering if your compounds have an effect on the subcellular localization of P53. I mean, some of your data suggests that it would, that you're moving P53 into the nucleus, but I wonder if you've actually demonstrated that.
We haven't looked at that specifically. We've looked at more markers of apoptosis and where those move, dependent on our treatments, but we haven't actually looked at the movement of P53 necessarily. We have the data, actually. We have the cells, we have the images, and so we haven't maybe analyzed in that way, but we could go and do that. But I have no information at this point.
I actually have a couple of other questions, but I'm going to forego them because we have to move on. Very nice talk and beautiful work.